Can I actually take this opportunity whilst I've got the mic to ask if we can give a round of applause to Rachel, Andy and the team from Reverse Rett for putting this day together. This sitting in the audience is all going to look seamless and everything will flow really very easily, but I don't underestimate how much work it's taken to get to this point. So thank you guys, and thank you for the opportunity to talk today. I'll fill a bit more time before the slides appear, but it's lovely to see so many familiar faces in the audience and actually to have the opportunity to meet some people like Jackie who've been such important people in the field that I haven't met before. And I've got my slides there, fantastic. So I do have just a few disclosures that I just need to acknowledge for the talk today. I think perhaps the two most important things are that I've done some consultancy work for Neurogene around their gene therapy trial, and as has already been said, I'm the PI for the TAISHA study here at the Evelina. So, my outline for today's talk, I always give an outline, I very rarely stick to it, but it's always good to have good intentions. I'm going to talk a little bit about what Rett syndrome is, my perspective as a clinician, talk a little bit about the link between MECP2 and Rett syndrome, and then obviously talk about some of the principles and challenges for gene therapy for Rett syndrome. Whilst I've also got the mic, the questions for the question and answer session are immensely important, so please do take the chance to write some down. There's no such thing as a bad question, and there are three types of questions. There's the questions we can answer straight away that'll make us feel really good because it sounds like we know what we're talking about. There's the questions that we'll think about and disagree, but there'll be good discussion. And the really important questions are the ones that no one can answer. But I think actually after a day like this, leaving and knowing what the unknowns are, I think there's real value to that as well. So, what is Rett syndrome? So the original description was from Andreas Rett back in the 1960s, and it's said to be the commonest genetic cause of learning difficulties in females. Definitions of Rett syndrome have changed over time, and the most recent definitions were back in 2010. I think they're due an update, but that's a discussion for a different day. And there's a very strong association with MECP2 mutations, but really importantly, it remains a clinical diagnosis. I'll say this a few times during the talk, but not everybody who has Rett syndrome has a MECP2 mutation, and not everyone who has a MECP2 mutation has Rett syndrome. And that's really important for the applicability of the clinical trials as we move forward. So who is Andreas Rett? There is a risk with medicine that we laud people with eponymously named syndromes, and it turns out they're not nice people. Fortunately, that is not the case for Andreas Rett. He is one of the good guys, and I think he's someone we can be really happy about. Number one, he carries off this sort of pale trousers, pale shirt look in a way that I could never hope to do that. Interestingly, so sadly he's no longer with us. He was disowned by his father for not pursuing a career in his family's cleaning company, but instead going into medicine. And what a different world it would be if he had chosen that path there. He graduated back in 1949, and he set up a lot of very good charitable endeavours, including a factory that was built to help support getting disabled youngsters into work. So he's a real, just as I say, an all-round good guy. His first description of Rett syndrome was in 1966. The original paper had about 22 cases in it, and it said that he came into his clinic room, and on one side of the room, and then on the other side of the room, he saw young ladies who were breathing differently, who were rubbing their fingers together, and a light bulb went off in his head, and he realised that this was a syndrome that he could describe. And I'll leave this up just to let people read it for a second. So I don't know anybody who's spent any time with young ladies with Rett syndrome who doesn't think this. So he's a good guy and he gets it. Yeah? He knew what Rett syndrome was about. So a gentleman I think that we should remember very fondly. So I've said that Rett syndrome is a clinical diagnosis. So what do I mean by that? I mean we diagnose it on the clinical features we see. So you have to have a period of regression followed by a period of then stabilization. That's one of the absolute criteria. And there are four major criteria that we see. During that regression, there's a loss of your hand skills, there's a loss of speech, there's the development of very characteristic midline stereotypies, then you go on even not to be able to walk or to have an abnormality of your gait. There are a number of minor criteria. I won't read these out because there's too many to read out there, but importantly, some of them are very specific to conditions like Rett syndrome, like the breathing disturbance there, whilst others are things we see in a range of other neurodevelopmental and other conditions. And importantly for the exclusion criteria, there has to have been a period of normal development before you have the onset of your regression, and there's no evidence of a brain injury or another explanation for it. And we talk about typical Rett syndrome or classical Rett syndrome. That's when you've got the four major criteria. Oh, is that projecting? Yeah, we'll go. And none of the exclusion criteria. Atypical Rett syndrome, you have to have two of these, but you might have an exclusion, but then you have to have quite a few of the minor criteria there. Now, anybody who spends a great deal of time with young people with Rett syndrome will recognise that this is an artificial divide and it's a spectrum of the disorder. But again, really importantly for our clinical trials, it's the typical or classical group that we're concentrating on. We'll talk about this a lot today, but you have to start with a very defined group of young people. That's just how all trials have to begin. So what is a gene? I'm going to talk about this in a very basic way, but the gene really is the basic unit of inheritance. So your genetic code, we can think of it as the recipe book for many of our biological traits and our physical characteristics. Now, it's not the only thing that defines how we turn out. Our environment is very important as well. And if we think about that analogy of a recipe book, it doesn't matter what ingredients we put into the cake. If we put it in the wrong oven at the wrong temperature, we'll get the wrong result there. So it's the interaction between our environment and our genes that make us what we are. Different genes provide instructions for making proteins. Back. But different genes provide instructions for making proteins, as Jackie's explained, and have different functions in different parts of the body. And we get copies of each gene. We get one from each of our parents, so one from our mother and one from our father. And that's particularly important when we're thinking about Rett syndrome. Now, these are our chromosomes. So our DNA is grouped together into 23 pairs of chromosomes. And as I say, you get one from mum and one from dad. And 1 to 22 look very similar. You can't really tell them apart by looking at them, but it's the sex chromosomes that are very different. And this is what they look like. And gentlemen, I'm sorry to say, but this Y chromosome is a pathetic excuse for genetic material. Just look at how pitiful that is. Ladies, you are strong, okay? You're so strong, you actually only need one of these chromosomes. So you've got 2 of them in each of your cells of the body, and one of them gets turned off. And that's really important for Rett syndrome because the MECP2 gene sits on that chromosome. Gentlemen, this thing just does its pathetic business in all of your cells and isn't very important, unfortunately. So if we're thinking about why a spelling mistake in a gene causes a problem, essentially it's because we either get too little of a protein, and that's the issue that we have with Rett syndrome. We can get too much of a protein, and that's a problem for our gene therapy, as we'll touch upon, or we get the wrong kind of protein that doesn't do what it should do. In very simple terms. So the aim of any gene therapy is to try to restore a functional level of a functional protein. So you have to correct that deficit by, in some way, putting back in a protein that's going to be at the right level to do its job and to do its job properly. So the MECP2 gene, we've already heard about this. It's the methyl CpG binding protein 2. I always take every opportunity to say it out loud because it took me a long time to remember it in medical school. So that makes us feel clever if we say it out loud there. And we know it is really strongly associated with Rett syndrome. So if you meet the clinical criteria for typical Rett syndrome, 95% plus of young people have that gene change. Atypical Rett syndrome, it's about 75 to 80, and there's a large number of other genes that are associated. As I've said, it's found on the X chromosome, and it predominantly, but really importantly, not exclusively affects females. And it's a really complex upstream gene, as Jackie's explained there. It's not a simple gene that's easy to replace the function of. And that's why it's so important that we're developing these gene therapy treatments. We've already heard that it's expressed in the brain and other body tissues, but it's the expression in the brain that's important. And that's very important for us in terms of how we deliver the gene therapy, as we'll talk about, because it creates some challenges, but it also makes some of the safety issues with gene therapy that Amy's going to talk about later today a little bit less of an issue for us, because there are unique risks to gene therapy, as it's really important as a community we're aware of. So the relationship between MECP2 mutations and Rett syndrome. I've said this before, I'll say it again though, not every individual with Rett syndrome has a mutation in the MECP2 gene, and not every individual with a mutation in the MECP2 gene has Rett syndrome. What's changed in the UK is our genetic testing. Now everybody gets whole genome sequencing, and I'm increasingly being asked to see young people in my clinic with a MECP2 gene that look very different to our traditional concepts of Rett syndrome. It's clearly a spectrum. This will emerge in the fullness of time. Now, I will gloss over this slide, don't worry, I'm not going to go through each of these pathways and explain, but again, it's just to make the point. It's complex what this gene does. It has many, many interactions, and that's why it's difficult to replace the function. So what are some of those functions? Jackie's already talked about this. but it has a role in neuronal maturation. So you don't lose brain cells when you have Rett syndrome, but the cells are a little bit more simple than they should be. They don't quite form the connections that they should, but that's potentially rescuable, as we've heard. It has roles in how we express other genes, how DNA is organised, and that has a downstream effect on our protein synthesis. And it's linked to a large number of neurodevelopmental disorders. So we've got Rett syndrome, MECP2 duplication, which is very important when we're putting the gene back in, as we'll hear, and then a range of other disorders. Now, this word neurodevelopmental, I think I've got a little bit of an issue about how we use this for Rett syndrome, because as Jackie's just shown us, if you turn the gene back on at any point in the mouse's life, the mouse changes. So the MECP2 gene is not just a gene that affects how the brain develops, it's necessary for brain function. And that's very important because it means theoretically, at any stage in a human's life, if we can reactivate the gene, we should see that there's benefits and improvements. And this is really important for we think in the future about how we roll out any therapies, because it's really important that everybody who can benefit has that opportunity to do so. So I'll gloss over this because it's been described far more clearly and eloquently than I can ever do there. But essentially we've got a complex gene, but there's two main domains that seem to give the function that we're interested in. This also doesn't project. This is the white mouse from Jackie, unfortunately. It's on my screen very well, but essentially it's looking at frequency of genetic changes, so how often you see it in young people with Rett syndrome, and where it is on the gene. And I want you to imagine that there's lots of spikes here, and not much elsewhere. So it's telling us that the mutations that cause Rett syndrome, they cluster around these two important parts of the gene there. So these are the bits that we have to prioritize replacing and getting back into the human. I've gone backwards. Right? That way. Okay. Now we've used these terms a couple of times, and it is something that I think you'll have come across when you're reading about genetic conditions, but I think it's important just to kind of spell out what we mean. So the genotype is your genetic characteristic. So when we're talking about the genotype of someone who's got Rett syndrome, we're talking about what the genetic change is that's happened to the MECP2 gene. The phenotype is what we see on the outside. It's how the human behaves, how the mouse behaves, their hair colour, their eye colour, all of those kind of features there. So it's how someone looks and acts. And if we look at this correlation between the type of genetic change, the abnormality we see in the gene, and the clinical severity of the young person with Rett syndrome, there is something of a relationship. So there is a link between the genotype, the genetic change that we see, and the phenotype, so how it affects young people. But there is quite a big overlap. And again, the more young people we test, the more we see these MECP2 mutations, the more that we realise that for any of these individual mutations, there's a big range of severity, there's a big spectrum we see. And that's very difficult for me in clinic if I'm asked to see a 2.5-year-old with a particular change, and the family say, well, what does the future hold for my child with this change? And I can't tell them. And I'm cautious about extrapolating for this kind of data for the individual. Equally, for our gene therapy, if we want to put something in early in the child's life, because it kind of feels like that might be the best, we don't know what was going to happen to that young person. So our lack of being able to predict early on what the future holds for a young person with any given MECP2 mutation is one of the big challenges we have in our clinical trials. So we said that gene therapy aims to change the phenotype by correcting the genotype. And one word that I'm very cautious about using is that word cure. And I think certainly from our kind of perspective as paediatric neurologists and clinicians working in this field, I think Amy, I'm putting a lot on Amy here for her talk later in the day, but she can rewrite it if she's not going to talk about this there. I think for us, the way we would approach this in our experience with fair conditions is we don't cure, we shift the phenotype very, very significantly. And again, that's really important because it means we need to continually improve any of the genes. If we get a gene therapy from these trials that work, we still need to be doing work to get better interventions. There will be other interventions that we need to have to continue to get the best out of them there. So it's phenotype shifting rather than cure, but in a dramatic, revolutionary way, we hope. So I'll go back to this. I'm going to remind everyone, men, hang your head in shame with this terrible bit of chromosomal material. But remember, young ladies have 2 of the X chromosome in each of their cells, whereas young men only have 1. And we talked about turning it off, this X inactivation. And really, if we have a group of cells and we randomly inactivate the X chromosome, about half of them will turn off the X chromosome from the mum, half of them will turn off the X chromosome from their dad. And, or in this case, the X chromosome that's got the genetic change on it. But if there's a skew to that, if you turn off more of the bad chromosome, you'll end up with a milder clinical picture. And if you turn up less of the bad chromosome, you'll end up then with a more severe clinical picture. So this is one of the big reasons why we don't see that really tight link between the genetic change and what the young person and adult looks like. It's because there's factors like this that also affect it. So, if we're thinking about how we approach interventions for any condition, the simplest way is to start by treating symptoms. So for young people with Rett syndrome who have epilepsy, it might be giving them anti-epileptic drugs, as Jay's going to be talking about later today. It might be talking about medications that help affect their breathing, but they're treatments that could work for any young person with that kind of symptom, regardless of what the cause of it is. The next step is trying to do something about the disease mechanism. So if you had a problem where an enzyme didn't work, replacing the enzyme there, correcting the mechanism in the cell or in the body that's gone wrong. And then of course the higher level is trying to change the genes to correct the defect, and it's at this level that the gene therapy is trying to work. Now over the last few years there's been a lot of trial work, a lot of interest in different medications and different therapies to help the symptoms of Rett syndrome. And essentially these are things that are working on the left on the symptomatic level, these in the middle are working more on the kind of disease pathway level, and on the right it's the gene therapy. Now, the problem of course is because it's such a complex pathway, it's difficult to replace the function because it interferes and it affects so many different genes and different protein synthesis pathways there. If we look at the Sarazotin, the trial from a few years back, this was very much a symptomatic treatment, trying to affect the breathing just by treating that as a symptom in itself. Trofinetide's probably more in the middle here, looking at kind of a more of a disease pathway. But of course, what we're interested in talking about today is this basic level, this correcting the gene defect, getting to the heart of the problem and trying to sort it by putting in a functioning gene. So, it's almost indecent how much I enjoy watching these videos of these mice here, okay? No one should take so much pleasure of the mice there, but I was an early stage registrar when I saw these videos, and it blew my mind. My wife had no idea why am I so excited about mouse videos, but I am. So I'm going to play them again even though we've just seen them, because I think it is just so wonderful. So look what happens. You turn on function. You're not moving and you're moving. This is amazing. In the field of paediatric neurology, to have such a clear change and such an improvement in the phenotype, this is dramatic. And the fact you can do this at any age of the mouse model, that is hugely important. So this is really, really, really exciting work. However, can anybody who is a mouse in the room please put their hand up? Okay, so we're not mice. This is a lovely hotel. I refuse to believe there is any mice in this hotel there, okay? But none of you are mice. I love cheese, but I'm not a mouse there, okay? And what we don't know is how we can extrapolate from the mouse experiment to the human phenotype, because the clinical picture is so much more complicated. So I know that when families come to see me in clinic, the things that they say are the biggest concerns for them, that would be the biggest priorities to improve, are communication and hand use. And these are things obviously which we see less of the mouse there. It's not to say that I don't think that these things can improve, but I think we need to do these trials, we need to do these experiments. So there is the question of, are these gene therapies going to work? But if they work, how much are they going to work? And what are the domains of Rett syndrome that they're going to shift? And this is the phase that we're entering into to begin to answer these questions, hopefully in the next few years with these coming trials. So if we think about challenges for Rett syndrome, and for gene therapy rather, there's a lot of things we can talk about. So we can talk about how we get the gene therapy into the brain. I'll just put these all up on the slides. I'm just going to talk very briefly about each of these before I conclude for today. So how about delivering the gene therapy to the brain? So we've seen that the important thing is correcting it in the nervous system. Doesn't matter so much where it appears in the mouse model, what happens in the heart or the kidneys, but the brain is what we need to target. Now, so we need then to get the gene therapy into the nervous system, and when it's in the nervous system, we need to get it into the cells of the brain, okay? And they're two things which are different problems we need to address in different ways. Oh, okay. When I had this on my laptop, this didn't look quite so much like a sandworm from June, so sorry, it's a bit scarier here. But there is a really important thing called the blood-brain barrier, okay? And people might have heard of this before. Your brain is kept very securely separate from the rest of your body, okay? And this is because variations in your sugar levels, variations in the salt that happen in the bloodstream, if they were to happen in the brain would stop functioning and you'd just fall to the floor and be sitting and kind of unfunctional the whole time. So you have a very complex series of barriers that prevent things easily going into the brain and out of the brain to keep it functioning. What that means is lots of medications that we give don't get into the brain very well, and gene therapies won't get into the brain very well. So if we were just to infuse a gene therapy into a vein, the amount of it that will get into the brain would not be as high as it could be. So we need to physically place it in. And that means we need strong arms and needles, I'm afraid. There's two different ways we can get it in, and these are how the trials are working. So one trial for the Neurogene trial is going to deliver this intraventricularly. Everybody has fluid-filled spaces in the brain. That's normal and healthy. And what they're planning to do is to put a catheter in, directly pierce the brain, and deliver the gene therapy to the fluid in one of the ventricle systems. Other way we can do it, if we're not going at the top end, is to go in the bottom end and to do a lumbar puncture effectively. So with the intrathecal delivery, the delivery into the fluid around the spine, you feel for the gaps between the bones in the back, and you put the needle through, and then you infuse there. Different approaches to breaching that physical barrier and getting the gene therapy into the nervous system, okay? Once it's in the nervous system, we have to get it into the cells. There are various ways that we can do this, but both of the gene therapies that Tasha and Neurogene have developed use an AAV9 vector, so a viral vector. So essentially, the virus takes the gene therapy into the cells of the body and it integrates it in the DNA. But of course the problem is, that's a virus, and the body doesn't like viruses. So that creates some problems that we need to address. So getting the right amount of the gene action, and I call this the Goldilocks principle, okay? So we know that if there's not enough of the action, not enough protein from the MECP2, we end up with the clinical syndrome of Rett Syndrome. We know though that if we have too much, we create a different neurodevelopmental disorder. We create a duplication syndrome. And whilst there are some features in common, there are differences there. The worst scenario for us as clinicians is what we do is we shift young people from this side of the slide to the other side. You know, that's not going to help anyone in the long term. How this is being got around is there's some very clever molecular mechanisms in each of the gene therapy constructs that are going to regulate the amount of MECP2 expression. Okay? So what it means is that it's not just getting it in, it's making sure that when it's in, it produces just the right amount of MECP2 to improve your function without causing these other problems there. It's very difficult for us to monitor for overexpression. The phrase people might talk or we might hear is biomarkers. This idea of something we could simply measure that would tell us whether someone's got too much MECP2 action, and there isn't a biomarker there. The data I've seen from the animal studies is really convincing. I think this is safe and appropriate, but this is one thing that we need to be monitoring for in our young people. So what about then ensuring the gene therapy is tolerated? So this is a foreign protein and it's a virus that's being put in, and the body will mount an immune response, and we need to dampen that down. Now, I'll look at Amy when I say this just to make sure she is going to talk about it, but there have been deaths in other gene therapies which have been related to the body's immune response to the virus that's been delivered. So I'll pause just to let that sink in, but the way we modify that, the way we reduce the chances, is immunomodulation. So you suppress the immune response from the body. That's not without its risks though. We're causing someone to potentially be more vulnerable to infection, a little bit like when they've had a transplant. There are lower levels there. The other thing is that blood-brain barrier works in our favour, because if it's difficult to get things in, once we've got them in, they don't come out very easily there. So from all the data we have from animal models, for either route of delivery, most of what we put in stays in the nervous system, less of it leaks out into the rest of the body to, of course, that immune response. But at some point, there will be problems. I don't know how many children, but it's a statistical risk, and this is a big risk for the gene therapy trials. Okay, measuring outcomes. We could have spent the whole day talking about this. It's really difficult to measure meaningful outcomes that demonstrate that this works and demonstrates that it's safe there. So what are the best measures to use in such a complex condition? How do we simply take the complexity of all the ways in which Rett syndrome affects our young people and actually demonstrate that that's changing, given that it's probably going to be very different for each individual child that we're seeing in the clinic? There's a large number of different outcomes that have been used, and I think all of us have our feelings of which are the best and which are the worst there. Some of them are things that the regulators now want us to use, so we have to. But for the early phase trials, there's throwing the kitchen sink, as it were, of all the assessments to make sure we're capturing the change. But it means that for the first phase 1-2 trials, the burden of assessments on families is going to be quite considerable. It's a necessary evil, but it is a really important consideration. How to balance, so I've already said that, so how do we balance the burdens there? And how do we make sure that we measure things that are important to individuals with Rett syndrome and their carers and families? You know, it's no good if I think it works, if it doesn't work from the young people and their families there. So it has to be measures that are meaningful to everybody that's involved. Okay, so I'll just briefly talk about this, but I'm sure we'll come later about it. But if we're talking about the phases of our clinical studies, the phase 1, phase 2 and phase 3. Phase 1 is the drug safe when it's given to healthy volunteers? But for an intervention of this nature, we can't give it to healthy volunteers. It would be completely unethical to give someone a gene therapy into the brain or into the spine with the associated risks where there's no possibility of them benefiting from it. So what we normally do then for the phase 2 is we combine that looking at, is it safe in young people or adults with the condition, and is there a suggestion that it's actually working? And then it's the phase 3 trials that then go on to demonstrate that something's working and it's worth being funded by the regulatory agencies. The Neurogene and the Taysha trials are combining these two. The focus is just making sure that it's safe and that there's some data, some evidence that shows that it's working, but the bigger trials are going to be needed to demonstrate that is the case and to convince regulators that they should be paying for this. So the last thing is understanding the natural history of Rett syndrome at an individual level. I've already talked about this. Sadly, when I became a consultant, I didn't get issued with my crystal ball, and I wish I did, but it's really difficult for us to know what the future for any individual holds there. And if the trajectory of change, if the individual path that each child is going to be on is different, how we measure that and capture that and show that there is a difference is I think, a real challenge. Now is the opportunity for us as a community to be collecting better data to use as the basis of the trials. And so we mustn't just be thinking about the trials that are here now. We've got to be doing that groundwork and that scut work for the future trials to make sure that we get these things off the ground. So we've used the word hope a lot today. And I think for all the challenges, for all the difficulties and the risks, there is a real sense of hope. You know? I am a relative newcomer to Rett syndrome, and you know, my sort of mentor, Dr. Hilary Cass, who ran the clinic at the Evelina, about 4 weeks before she retired, she said, "Dan, you're taking over my Rett clinic." So I had a crash course in learning about this, but by God, what an amazing opportunity and time to come into the Rett world there. This is a real sense of hope and opportunity, but I deliberately have picked a picture of hope written on a stone because stones sometimes sink. And that sense of hope can drop away at times. And I think standing here at the start of this journey, we've got to recognize and realize that there are going to be upsets, there's going to be hiccups, there's going to be problems, and there's going to be setbacks. But we've got to keep holding on to this. And it's that winding road, isn't it? And I think, you know, we're about maybe there now. We're going up, by the way, we're not going down. So it's not almost over, but there's a lot of curves, there's lots of things. And the problem is, when you're here, you don't quite know what's around that corner because you can't see it yet. This is an amazingly exciting and hopeful time to be involved in the Rett world, but we've got a lot of challenges. But it's organisations like Reverse Rett, it's bringing people together, it's days like this and the sense of community it creates that I think are going to stand us in really good stead. So I'm going to end there, but I always put this slide up. I think I'd be amazed if anyone in this room isn't signed up to the registry there, so it's probably not a slide I need for this audience. But if we're thinking about what families can do and the single thing that will help us the most, it's registering their children and adults with Rett syndrome. We need to know where people are, we need to be able to collect this data, and that's going to be immensely important for trial design, and it's also going to be very important for regulatory approvals moving forward. Okay. So I'm going to stop there because we've got an opportunity to talk a little bit later, but thank you everyone for your time and attention, and thanks to the team at Reverse Rett for inviting me today.